welcome to the Disrupting PFAS podcast. I'm your host, Jeff Hale. For this season of the podcast, our theme switches to natural materials and processes for the detection, destruction, and sequestration of PFAS. Today, I will be talking with Dr. Peter Jaffé of Princeton University about biodegradation of PFAS. I chose this topic because it challenges the notion of PFAS as forever chemicals, and the prospect of PFAS biodegradation is desirable and encouraging. So let's learn more about this topic from one of its innovators. Welcome to the podcast, Peter.
Hi, Jeffrey. It's a pleasure to be here. Thank you. Thank you for inviting me.
You're welcome, and we appreciate you joining us. So I guess just so we can get acquainted and the audience can get to know you a little bit better, could you please tell us about your position as the William L. Knapp 47 Professor of Civil Engineering at Princeton?
I'm a professor in civil and environmental engineering at Princeton University. I'm also a member of the Andinger Center for Energy and Environment and the High Meadows Environmental Research Institute. My background is in water pollution. I've been looking at fate and transport of contaminants over the years, looking at both physical, chemical, and biological factors that affect the fate and transport of contaminants. Focused both on surface waters, groundwaters, groundwater remediation, and also wastewater treatment. I do both modeling and experimental work and have often liked sort of the interface between modeling and experimentation, use experiments to drive models and models to drive experimentation. And I've been at Princeton since 1985, so pretty much all of my career.
Okay, great. Well, fate and transport of contaminants is definitely a passion of mine. Also interested in connecting models with empirical evaluation. So I'm glad we're able to have you on the program to talk with us today. You mentioned physical, chemical, and biological processes. That's something also that's interesting to me, that multifaceted research approach. Could you talk a little more, please, about how you combine physical, chemical, and biological processes in your research approach?
Yeah, I mean, in a sense, when you look at the fate of a contaminant in soil or water, surface groundwater, you cannot separate them out. I mean, when we look at, very simple, at the biodegradation of a component, sorption is important. In general, we believe that the strongly sorbed component is not easily degradable, that it has to desorb first before it's degradable. So in order to understand biodegradation, we need to understand sorption. Many organic contaminants, volatilization is important, so we need to understand the partitioning between gas and water phase. We've been looking at contaminants in wetlands, where flow of water in the rhizosphere and the flow towards the plant affects the flow of the contaminants in the rhizosphere. You have pockets from a chemical point of view that are sort of sometimes quite interesting, where you do have pores that are highly reduced, others that are more oxidized. So we can come up very simply with a average redox potential, but that average redox potential doesn't really exist. We have this binomial distribution. When you look now at the behavior of the chemical at that average redox potential, it's doing something completely different than what you observe. So you do have to understand the spatial distribution of, let's say, redox potential in soil, especially unsaturated soils, sorption, volatilization. So it's hard to look at any of those problems holistically without combining biological, physical, and chemical, all three of these processes.
Mm-hmm. Well, that makes a lot of sense, and I'm interested to get more into, PFAS in particular, where it occurs in these environmental compartments or different parts of the environment, as well as some of the redox conditions. But before we get into that, I understand that you spoke about PFAS to the U.S. House of Representatives, subcommittees on the environment, and also research and technology. I guess that was back in December. So first of all, thank you for representing the science of PFAS in Congress. And would you tell us about that experience, please?
Sure. The objective of that committee was to identify research needs to understand the fate and transport of PFAS, identify where the federal government could help in aiding research and research collaborations between agencies, etc. Two sort of broader issues that I brought up was one that, well, the latest I looked, we have 4,700 PFAS. I think later numbers show over 9,000. I'm not quite sure where we are. That's a huge number, and the properties are very, very different. It's when we looked at PCBs, for example, 30, 40 years ago, the number is much, much smaller. I think the typical different monomers are somewhere in the 130 range. Their properties are very similar. When you look at fate and transport of those different PCB monomers, they're not terribly different. They're not as varied as PFAS. So we cannot study 7,000, 9,000 PFAS individually. We need to group them into groups that have similar properties and then understand their toxicity and transport based on those properties. For example, we can focus on perfluoroalkyl acids and then see systematically what happens as the carbon chain becomes longer or shorter. So one recommendation was to sort of group them in a logic way and understand the transport toxicity of them by group. The other sort of broad recommendation plea I made is that a PFAS analysis, as you know, is extremely expensive. When you send out a sample for commercial analysis, we're talking about $400+. That is a barrier for many individuals to enter PFAS research. So there could be lots of scientists that have good ideas on what to investigate, but the analytics is a barrier. So there are multiple ways of addressing that. I'm not an analytical chemist, but can we develop analytical techniques that are not quite as expensive? In the absence of that, if we're talking about federally funded PFAS research, there are Department of Energy examples where we have large analytical facilities, user facilities. The PFAS user facility may be something that would facilitate much research in the PFAS area. And finally, if none of those is feasible, Congress has to realize that doing PFAS research is more expensive. That NSF, the typical NSF grant is 100,000 a year. Maybe you can do it when you focus on nitrogen or TCE, but not on PFAS. So the grant has to be proportionally larger to allow for these costs. So that is sort of the larger examples. More detail to PFAS, I sort of point out that we need to understand sources. We don't fully understand sources. We have focused on the AFFF sources, firefighting foam. EPA is focusing very much on landfills, which is a big source of PFAS. But what else? We know that there's some agriculture fields or cattle pastures that are contaminated with PFAS. The milk has to be thrown away. We don't understand these sort of kind of more diffuse sources. The Cape Fear River has large sections of sediments contaminated with PFAS. We are doing research on the Neuse River. We see PFAS in many locations in Neuse. That is very little understood. And so we need to understand those more diffuse sources. How important are they? Can we address them? Then sort of digging down more, like going for example, at my interest level, we know that PFAS can be biotransformed, and there is limited evidence that PFAS can be degraded, that perfluoroalkyl acids can be degraded. We need to understand that at a very fundamental level. What the organisms that are able to degrade PFAS, what are the key enzymes? What are the genes that are being expressed? can we use that to track in other environments? Is that gene present or some similar enzymes present? So that, and what is needed to stimulate it? So that we may have organisms that could defluorinate PFAS, but they need something, some specific redox potential or very specific conditions that we don't understand. And for bioremediation, so we need to understand that.
Well, I think that's great perspective. I think that that captures a number of key elements of the PFAS issue. And again, appreciate you, representing the science of PFAS to Congress. You mentioned the perfluoroalkyl acids. I think, that's obviously a natural grouping to look at. And I think that's one that we're probably most aware of and taking head on, but, maybe there are other groupings out there. So in your answer to that question, you also started to get into kind of crux of your research, and that's the microbes that might be able to degrade PFAS, and that's what I wanted to get into next. So I think that's a good segue. I understand that Acidimicrobium bacterium A6 is the microbe that has been identified to degrade PFAS. So I guess first of all, did I pronounce that correctly?
Yes.
Okay.
A mouthful, yes.
Okay, so I guess first of all, how was that microbe isolated and identified?
That's an interesting story, and let me go back. So we did work in the early 2000s, so 25 years back, on, and much of my work focused on wetlands, biogeochemical processes affecting pollutants. I was working with a colleague who unfortunately is no longer with us, Joan Ehrenfeld from Rutgers. She was a wetland ecologist, plant ecologist, and we were looking we had a USDA grant to look at the effect of nitrogen runoff into wetlands. She was interested how it affects plant speciation. I was more interested how the nitrates affect the iron cycle, since nitrates, especially nitrites, can reoxidize ferrous iron, and that then would sequester trace metals or metalloids like arsenic. So I wanted to see how that affects these dynamics. We did the preparation. We did some field work at the Assunpink Wildlife Management Area in New Jersey. Year one, we had our plots, reference plots, that the next year we wanted to fertilize, and the next year was a drought. So you could have had a completely dry picnic in your wetland. So out of desperation, I told my student, Juno Shestra, let's take a few soil cores and take them to the lab and incubate in the lab. That's the next best thing we can do. And then we can add ammonium or whatever to, and that was fortuitous, but that yielded our findings. So when we did those incubations, Juno came to me and says, look, ammonium is going down, and ferrous iron is going up. And so I said, look, I have, besides Anamox, I mean, we believe that ammonium is mostly an aerobic oxidation process, and Anamox requires nitrite. We did not have nitrite or nitrate in the soils. So as the first test is, let's do the energetics. Is it possible that you could oxidize ammonium with ferric iron as an electron acceptor? And yes, the thermodynamics showed that it is feasible. So we published in 2005 a little paper that sort of postulated that we have ammonium oxidation under iron-reducing conditions. A year later, completely independent, a Japanese chap, Sawayama, found something similar in a reactor, and he called this process Feammox as a play to Anamox. And the paper is not very detailed. You don't know the redox potential in its reactors, but okay. So his contribution was this name Feammox. Then again, maybe 2012, a group at Berkeley found something similar in rainforest soils in Puerto Rico. And then we got sort of really our interest peaked more and says, look, this is something. There's something to it. I'm fortunate being at Princeton. We do have some internal fundings. It's not trivial for an engineer to go to NSF and say we want to dig into microbiology. We think there's a new pathway in the nitrogen cycle. We get never fly. But so we got some internal funding. I hired a microbiologist, Dr. Huang. We went back to the site. We were delighted we could find our site 7 years later. We were delighted that we could reproduce our results from 7 years later. So we could do the incubations. We did see under anaerobic conditions iron reduction and then the production of nitrite and ammonium disappearing. And then with very careful work, Dr. Huang managed to detect that there was an Acidimicrobium that was growing, and over the 100 years incubation, that culture was enriched for this Acidibacterium. And the name A6 is sort of fortuitous. She had multiple experiments, A, B, C, D, 1, 2, 3, 4. And the group A and the sixth one is where she found this organism growing. And I think we could have probably come up with something more creative in naming it, but that's how it was an Acidibacterium, and that's how the A6 came from. So we linked that to the Feammox process. To be honest, it was a major pain to get that published. Microbiologists were a little skeptic of engineers coming up with a new organism, but we got it published. Then we went further once having that organism. So again, we linked it to the Feammox process, which is ammonium oxidation. Ammonium is the electron donor, iron, ferric iron is the electron acceptor. But we couldn't really prove in an enrichment culture that that organism was the one responsible for the Feammox process. In 2018, we managed to isolate it and show that the pure culture of this Acidibacterium A6 was able to oxidize ammonium and reduce iron. We then looked at the sequencing, and we found that it had a series of interesting genes for enzymes. One was enzyme very similar related to methane monooxygenase, and that one was expressed every time we had ammonium oxidation. The organism can either at least live with ammonium as electron donor or hydrogen. When it grows on ammonium, it expresses that enzyme, that gene. When it grows on hydrogen, it didn't, and actually over a few generations it would lose that. It was that gene is on a plasmid, it would lose it. So going back to TCE, we know that trichloroethylene can be epoxidized by methane monooxygenase, and since this was related to methane monooxygenase, we tested and we could see that TCE was degraded. That was a little paper. Then we looked more and we realized that it has a whole bunch of dehalogenases. So the next question was, given the interest in PFAS, can we degrade PFAS? So I talked with Dr. Huang and we said, look, let's get these. I think we picked 15 different PFAS and let's do incubations. Let's see first at what levels they're toxic. Most were not toxic. In most cases, we could go to over 100 milligrams per liter, and they were not toxic in a sense that it would not suppress significantly the rate of ammonium oxidation.
Okay.
So given that we didn't have the analytics, that looking for fluoride is easiest, we did a whole bunch of incubations, all 15 of them at 100 ppm. And sort of 3 weeks later, she knocks on my door and says, I see fluoride. And I said, in which one? And she says, in all of them.
That's exciting.
That was exciting, but of course, exciting, but I did not believe 100%, right? So the poor Dr. Huang had to do things over and over, and we sent it to multiple labs for independent verification. But it always came back that we did defluorinate these PFAS. Then we focused on PFOA, PFOS. Again, we did much more careful incubations, and we sent them to different commercial labs. They all came back the same. Fluoride analysis are not trivial. So we did the fluoride with different ion chromatography columns. We did send some out analytically, but at the end, we convinced ourselves that this is real. And that was then the paper that we had in ESNT in 2019. Again, sort of, we tried, I think, Nature, and they said we're not interested. Then we tried PNAS, and they said, oh, this is a topic that is not of enough interest. So we went to ESNT, and the paper has over 24,000 downloads so far. So there is interest in the topic, as you and I know, but the editors did not necessarily see it. We have since looked more carefully at multiple PAs. So you know that especially the hardest PFAS to defluorinate are the perfluorinated ones that are fully saturated with fluorine except the function group. So we tried several polyfluorinated alkyl acids from 10 to 4 carbons. We could degrade them all. Puzzling still, and I don't that the perfluoropentanoic acid was slower than the hexanoic and butanoic acid. So, but different vendor, so we don't quite understand why there was this dip for that specific one. What was interesting, we then looked at several of, well, no, we looked perfluorooctanoic acid and perfluorooctanosulfonic acid, and we looked at linear molecules versus branched ones. And branched ones are not easy to get. We typically have to get analytical standards for the branched ones. And what we could see is that the linear ones, we could defluorinate a single branch, we could defluorinate 2 branches would be inert. So we would not, somehow defluorination didn't work. So I think the majority of the perfluoroalkyl acids are linear, but there could be a few branched ones. When I talked to folks at Chemours, they said at least what they produced originally they believe was mostly linear. It is possible that the branched ones, branched perfluoroalkyl acids, are products of the degradation of compounds in AFFF or other kind of precursors. So that's where we are. This Actinobacterium is a real, I mean, as exciting as it is, it's not your favorite bacterium. Your E. coli has a doubling time of 20 minutes. This has a doubling time of 2 weeks. So it's and you can imagine, I mean, it's anaerobes grow slow, autotrophs grow slow. Here we have an anaerobic autotroph. It grows agonizingly slow. And that is something we need to work with. I mean, if you, as someone working or interested in bioremediation, you could conceive that we would want a reactor that we grow big culture of these organisms and then inject them into a contaminated site. And of course, that will make things much more challenging. The other challenge is, and we have been able to grow enrichment cultures in membrane reactors, but it needs iron, and the stoichiometry is 6 iron per ammonium, right? So you had that reactor, you had to add a lot of ferric iron, and after a couple of months, you have a solid mess, and it's very hard to separate the bacterium from your ferrihydrite. The ferrihydrite gets slowly converted to magnetite, but we haven't figured out a good way to separate them out. Fortunately, we have seen that you can grow it in electrolysis cells. So instead of transferring the electron to ferric iron, it can also transfer it to an anode. And we are now trying, and it's touch and go, to grow it in a continuous way in bioelectrochemical reactors so that we can grow it in the absence of iron. It's not working 100%. It's could be mechanical issues with our reactor, and I never know if we're 2 weeks from a breakthrough or 2 years. So it's a little frustrating. But the organism is present in many soils. So the organism is it's not it's we have done a survey. That was a paper, I think 2016, where we surveyed a whole bunch of soils in New Jersey that we just took a bunch of samples, and then we collaborated with the Chinese Ministry of the Environment. They sent us hundreds of samples from Southwest China, and we could show that the organism is fairly common in iron-rich acidic soils. They have to be somewhat waterlogged. It's anaerobic, sort of same as geobacterium, and you need this. It doesn't have to be saturated, but you need a moist soil to see them. So you need some pockets that are anoxic. We did get a sample from McGuire Air Force Base. We did some initial DNA extraction. We didn't see it. Then we just, and there was enough ammonium. There was no ferric iron, only ferrous iron. So we added some ferrihydride, let it sit for 2 weeks, and it was there. And we could see that once it started to pop up, we could see changes in the composition of PFAS, because it was a PFAS contaminated site. But there were too many precursors. So all we could say is, yes, once we stimulated it and added ferric iron, we saw changes in the composition, versus the samples that were sitting there that were not stimulated with ferrihydride. That tells me that even if the organism is slow, that there could be some sites where we can stimulate it so it starts growing without having to inject it. Probably acidic iron-rich sediments, but at least there would be some locations that we could start.
Do you think there are instances where you have the occurrence of PFAS, you have the occurrence of the A6 bacterium in those geochemical conditions that you described, anaerobic, iron-rich, and wet? I mean, that's a lot of variables that have to come together, but do you think there are instances out there where these microbes have been working on PFAS, perhaps without our knowledge? Or is it something that would have to be stimulated
No,
to be useful?
I think there's a good chance. So we have been looking at the Feammox process in the Neuse River in North Carolina. And the reason we focus on that is that along the Neuse, there are lots of these concentrated feedlots. There's a huge amount of ammonium going into Neuse. When you check at the concentration of ammonium, it's about 5 milligrams per liter of ammonium in some locations that we sampled. The Neuse has in general, acidic pH, 5-ish. And that area is iron-rich. The soil is iron-rich, lots of orange clay when you drive around. So we thought that may be a good location to find the organism. We did some survey, we found it. We did incubations, and we saw that there was a lot of ammonium oxidation under anaerobic conditions. And then we decided, let's track some PFAS. So we did two things. We added a little bit of PFOA. We saw that there was defluorination. And then we tracked some sediments that we just incubated without doing anything, analyzing for PFAS at the beginning and at the end, and we tracked PFOA, for example. We could see that it seems to be going down in some of those incubations. Now, of course, we disturbed the sediments, right? Of course, we shoveled them out, had them in a bag, put them in the lab, put them in an incubator. So we didn't add any additional chemical, but did we somehow make iron more bioavailable by transporting them? I don't know.
Well, you can't sample anything without disturbing it or measure it. That's the nature of science, right?
But these results indicate that it is possible that there are natural conditions where PFAS are actually being defluorinated.
I mean, that's exciting, and definitely one of the reasons we wanted to have you on today. That 2019 paper that you mentioned definitely piqued my interest, and I think it piqued the interest of a lot of people who had been holding out hope that there might be a biodegradation pathway for PFAS, including the perfluorinated compounds. So, Dr. Jaffé, I understand and appreciate, there might be challenges ahead to, optimize this bacterium and how it treats or degrades PFAS. But do you think is it possible that this is the tip of the iceberg, that maybe there are other microbes, bacteria out there in nature that we just haven't identified yet that could be capable of degrading PFAS, or may actually be in the process of doing so in the natural environment?
I'm optimistic that there will be more organisms that are able to degrade PFAS. And we may be at a point where we were. I wasn't part of it, but 40 years ago with chlorinated contaminants, right? We thought they were not degradable, and then they found some organisms, and then there are plenty of organisms that seem to be able to dechlorinate different chlorinated compounds. Of course, chlorinated compounds are more common. They're more natural analogs than fluorinated compounds, but there are fluorinated compounds in nature. Having identified the dehalogenase gene, that is the gene for the dehalogenase, the reductive dehalogenase that's expressed every time we see defluorination by Acidimicrobium A6, you can do a search, and there is another Acidibacterium that was discovered, I forgot the name, in the Black Sea sediments, 2018 or 2016 or so, that has the same sequence for dehalogenase. So other bacteria seem to have the same sequence for the same dehalogenase, and then there's no reason not to believe that there couldn't be other dehalogenases that could defluorinate PFAS. There are some indications that some Pseudomonas, some initial results may have been linked to defluorination of PFOA. It's very incipient, these results. And there are folks that are working on fungi that seem to also indicate that there's some fungi that can defluorinate perfluoroalkyl acids.
Okay.
I am optimistic that in a couple of years there will be more discoveries and hopefully organisms that are a little bit less finicky than this Acidibacterium.
Yeah, I think there's a lot of hope and optimism for a biodegradation pathway for PFAS. I think you do a good job of keeping it in perspective and, articulating, the challenges ahead and what needs to be done. But it just, I just think back through history and other chemicals and contaminants that have been a challenge. You mentioned chlorinated compounds and PCBs, and it seems like as history tends to repeat itself, eventually there's a bacterium that can degrade it, and people find it to be occurring more and more in nature. But you also make a good contrast between PFAS and some of these other compounds. You started out by saying, PCBs are so fewer PCBs than PFAS, and the chemistry is different. So I think that's important to bear in mind. I guess maybe working towards applying or relying on these bacteria for a solution, I wanted to switch gears a bit and talk about biosolids. Congratulations to you and your team on your recent publication in the Journal of Hazardous Materials, and that paper is titled Anaerobic Degradation of PFOA in Biosolids by Acidimicrobium sp strain A6, the bacterium that we've been talking about. And as I started to mention, the topic of PFAS and biosolids has been receiving a lot of attention in the scientific and regulatory communities. So I think the prospect of practical application of PFAS biodegradation and biosolids is encouraging, and also tying it back to some of our original dialogue, Dr. Jaffé, you talked about we need to understand sources better. And we're very familiar with firefighting foam sources, but there are some more diffuse and non-point source type sources where, for instance, biosolids have been land applied. So I think that that's a good example of those types of sources. So with that, could you talk about that study and some of the results, please?
Sure. Yeah, look, to be honest, I hadn't spent this much time thinking about biosolids until I got a lot of phone calls from plant operators, industrial and domestic, different parts of the world, after our 2019 paper about would it work in biosolids. So our interest got piqued, and we decided to look at biosolids, and then we started a collaboration with Chemours. They have a plant. They have plants where they had PFAS. They were generous enough to provide us their sludge. We got biosolids that we generated in the lab. We wanted PFAS-free biosolids, so we could do some kind of a mass balance. And so we generated biosolids in our lab and then contaminated them with PFAS. And the question was then, once we have the biosolids, can we seed them with the Acidibacterium A6, add ferrihydride, and would we degrade the PFAS? We focused here on mostly PFOA. And one of the questions was, when you look at sort of the traditional way you write the degradation of a sorbing component, you see that the rate is then divided by 1 over the partition coefficient multiplied by the density. It's somewhat reaction retardation. So I was afraid that maybe that would slow the reaction very, very much. But we did the incubations, and we could see that the dissolved and total PFAS in the biosolids augmented with the bacterium, and only if we added ferric iron, we would see defluorination. And that happened in our clean biosolids that we contaminated with PFOA, plus the ones that we obtained from the industrial plant. The real field ones were a little bit more complicated, and we saw that there was, I think it was perfluoroheptanoic acid, if I remember right, you have to look at the paper, was actually increasing, indicating there was some kind of a precursor, but PFOA, perfluorooctanoic acid, was decreasing. We saw the production of fluoride, and we could see that it was not just the dissolved PFOA, but also the total extractable PFOA in the biosolids were decreasing. Again, at a rate, I have to look at the paper exactly, but half-life of a month roughly or so. It's not shot terribly fast. I mean, but it was measurable. So the next thing we need to look at is how can we scale that up. And so now you could conceivably what we have in mind, and that would be, let's say, if we have a pile of biosolids that are contaminated with PFAS, can we add a ferric iron source, seed them with the Acidibacterium A6, let them sit in a pile for a month. Hopefully the PFAS goes below a desired level. Then maybe we can dispose 2/3 of the pile and leave the other third as the seed for the next round. That's what we're doing now in the lab. With sludge from our local treatment plant. Right now, the very, very first results, the Acidibacterium A6 is not growing as well in that sludge as we would like to. But we just started. Now, it would be, if a concept like what I just described works, it would be a relatively simple way of treating or removing PFAS from biosolids.
Starting to wrap up here. we really appreciate your participation and going into the details of the Feammox process and how these microbes can degrade PFAS under certain conditions. So Dr. Jaffé, could you just sum up for us the challenges, limitations, and further research that needs to be conducted or that you would like to conduct?
We have, because of again analytical difficulties, we find it easy to work with somewhat higher concentrations. We do not know for certain how low we can go in terms of degradation. Once we are in the parts per trillion level, can we bring it down to drinking water standards? I'm not terribly concerned if we couldn't. I mean, if we focus on highly contaminated source areas, and if you can bring it down by an order of magnitude, that's a big win. But so how low we can get at this point, I don't know. When you look at all of our experiments, we typically remove 50%, but we can always remove 50%, independent on the concentration we started. And at that point, we stop removing ammonium, right? So in our little batch experiments, we are building up enough metabolites that will and degradation products, ferrous iron, pH goes up. We consume the CO2. It's an autotroph that needs CO2 as its carbon source, so that the reaction will stop. So I don't think that any of these would indicate that we cannot go further in terms of that there's a lower limit of PFAS. It's just that the reaction, even without PFAS, even the simple ammonium oxidation stops.
Okay.
How far? So we need to understand how to go from these sort of laboratory batch experiments to something that mimics nature a little bit easier, and that's not easy to do something anaerobic, well controlled, but in a continuous phase. We have to do, and I hope that we will be able to do some field testing. We are getting certain funding to identify sites and identify conditions, so that maybe after that we can try to do a little bit of go to a field testing site. Hopefully one where the organism could be available. The next challenge is, even if it is available, even if it exists, if you stimulate it a lot, you're going to run out of ferric iron, right? And it needs ammonium or hydrogen and ferric iron. So ammonium is easy to supply, but ferric iron is not. It's a solid, right? And it's positively charged. Our soil is negatively charged. So how do you supply it to an aquifer? I mean, the sludge is easy. We just mix it in. Topsoil may be easy. You can plow it under, but once you go a little bit deeper than plow depth, I mean, how do you supply that ferric iron? So we are looking at ways of encapsulating ferrihydride in different polymers and change the zeta potential to make it transportable. So far, results are promising. We can do that as long as divalent ions are not too high.
Well, we hear a lot about the recalcitrance of PFAS and the strength of the carbon-fluorine bond, but it's interesting to hear that the limiting factors here might be the availability of ammonium and iron especially. So that's interesting. Well, I think that sums up, the challenges in the work that needs to be done, and I think people need to. I think everyone's excited about the prospect of the biodegradation of PFAS, but we need to keep it in perspective. And I think you've given us good perspective on that. But let's conclude on a positive note. And could you outline what are the exciting prospects, and what could the future hold for the biodegradation of PFAS as a solution or part of a solution?
I think that for at least some sites, we will be in a few years at a point where we can stimulate organisms, bioaugment them if needed, to degrade PFAS, and at least reduce the mass of the perfluoroalkyl acids to levels that are more acceptable. We can hopefully also understand under what conditions in nature there may be natural defluorination. I think our examples in the news are promising, and we much more preliminary. We in the Cape Fear, which is right next to News, we don't see it so far, but there Cape Fear has a higher pH than the News, so we may be able to identify what environments you are more likely to see natural attenuation.
Well, I'm definitely looking forward to learning more and hearing what those environments might be. So with that, that wraps up this episode of the Disrupting PFAS podcast. Thank you to Dr. Peter Jaffé of Princeton University for joining us today. It was a great conversation and great information. I'm your host, Jeff Hale, reminding you to never say forever.